get a doubt like how bacteria can be observed under microscope. There is a process called staining. Staining process is there for bacteria to observe under microscope. Here, these are the bacteria cells, different shapes. Some are rod shaped and some are circular. And these bacteria, when stained and observed on the microscope, you can find them in two different colors. Either they may be dark red or they may be purplish violet. This whole process requires you to understand a staining of bacteria by a method called gram staining. Bacterial cell, how usually you will observe. As of we are saying, there will be of different shapes like rod shape, circular shape, and they show some characters, how all these things can be known. For this process, there is a method called gram staining. Says you'll be having a glass slides like this. Transparent glass slides. Okay. You have to form a smear on this glass slide. This is the first step. This is the glass slide. You have to form a smear. What does mean by smear? I'll tell you. Just a simple one. Smear is nothing but take a drop of water or take a spit of your saliva. Just spit a saliva on this glass slide, just a drop. That is also a smear. Smear is nothing but the one which you are going to observe in later under microscope. Now you have to fix the smear. How you will fix the smear? For example, you have taken a water drop on this glass slide. You will be having Bunsen burners in your lab. Okay. Now Bunsen burner and light up the Bunsen burner, and above the Bunsen burner keep the slide here and there, so that this water drop will dry, heat dry, and get fixed on this slide. Okay, this is the first step. Then, what you will do in the next step? In the next step, you are having a slide with fixed water drop or a drop of your saliva. Anything is okay, not a matter. Now, to this you will add some chemicals. Very simple chemicals every lab will have. First chemical is crystal violet. First add few drops of crystal violet onto this and wash under tap water. Then add iodine solution. Few drops of iodine solution on this. Flush under tap water. Then add alcohol, the ethyl alcohol or acetone also to this slide and wash off. And at last add saffronin. All these chemicals would be available at any lab. Even in chemistry lab also all these chemicals would be available. So these are the steps. First add crystal violet, just to drop, wash it under tap water. Nothing will happen to the smear. You fixed it now, heat fixed it. Then add iodine drop, wash it under tap water. Then add alcohol or acetone, anything is okay. Wash it off. Then add saffronin. 1 drop is enough. Add it and just wash away. Now take a tissue paper. Next step. Take a tissue paper. Just put the tissue paper on this and remove the tissue paper. Don't wipe it. Just keep it on the slide and take it out. Now take this slide and observe under microscope. Nowadays we are all using compound microscope, but in some schools simple microscope is also available. If you go for further studies, you can have an availability of electron microscope also. Now under microscope, keep the slide and observe under 10x. You will be having under the lens, beside you will be having 3 knobs. 10x, have you observed 20x, 100x like this, if it is a simple microscope. This is nothing but the resolution. Resolution of the microscope, the detail of the microscope lens. We call it in these terms. 10x, 20x, 25x, 100x, something like that. If you observe this slide with the smear, after all these steps, either this you will observe, A, like this, or like this you will observe. These are the slides. These are called slides. Okay. So here, the actual bacterial cell can be observed either in red color, dark red color, like this. So by observing the shape, you can say these are of which shape? These are of rod shape, isn't it? Rod-shaped bacteria. Okay. Now, rod-shaped bacteria in one slide, they may be observed as red color. If red color is not observed, you may observe this purplish violet color, rod-shaped structures. Either this or that you will observe. Based on that, you will call as gram-positive bacteria and gram-negative bacteria. Actually, the question was very simply asked, gram-positive and gram-negative staining is related to, that's all. This stain, here we are using different colors now, like crystal violet, saffron, all the colors we are using. That's why it's called staining. And Graham is the scientist who contributed this work of observing bacteria under microscope. You cannot observe bacteria under microscope simply by having a liquid drop, something like that. You won't observe anything. The water drop only you will observe under microscope, as with naked eye. If you want to see bacteria even in water drop, even in your saliva, even in soil particle, you have to go for the staining process. This is the Gram staining, which you use to observe the bacterial cells under microscope. Based on the color observed, they are called Gram positive and Gram negative. Here, if you take the Gram positive bacteria, the gram-positive bacteria will be in violet purple color. This is gram-positive. Okay. Then gram-negative bacteria, they will be in red color. This is gram-negative.
Then bacterial cell.
Yes, bacterial cell. Gram staining is related to bacterial cell. Okay. One more question. I want to answer from another student. Gram staining is used to observe bacterial cells under microscope. The agents used in this process. First one, crystal violet. Second one, iodine solution. Third one, saffron. Fourth one, all the above. Which option will you go for?
Ma'am, all of the above.
Yes, Bhumika, yes.